Okay, to get ready for your first big lab practical, your first big histology practical, these are the first things that you need to know. You're going to be asked to identify what you see through the microscope and then to classify it. Tell me one location where you'll find that's something in the body and give me a function for it. So for instance, if you look through the microscope and you see simple squamous epithelium, you'll put that on your paper. That's your identification. And then you'll classify it as one of the four tissue types, as epithelial tissue, as connective tissue, as muscle tissue, or nervous tissue. Well, simple squamous epithelium, that even has it in the name, is epithelial tissue. So if you saw that in the microscope, you would say, epithelial tissue. And then you would give me one location where you find simple squamous epithelium in the body, and for instance you would find that in the lungs, so you would write down lungs. And then you'd give me one function. Now simple squamous epithelium is found in more than one place in the body. Don't write down two places. For instance, you can find it in the lungs and in the kidneys, but what if you put down lungs and you blank and you put down spleen and you don't find it in the spleen? Then you're going to be in some kind of trouble, because I'm not going to give you half credit there. I'm just going to count it all wrong. And same thing for function here, only give me one function. Simple squamous could be for diffusion and filtration, but what if you put for diffusion and some other function that doesn't count? I'm not going to give you half credit. I'm going to count it all wrong. So don't get fancy with it. Just give me one function, please. Okay. Now if we could just get my little things to show up. Good. Now in this lecture we're just going to go over the epithelial tissues, and we're just going to go over epithelial tissues. And for your epithelial tissues, the best thing for you to realize is, first of all, you've got your cell shapes. You've got squamous, which are the squashed cell shapes. You've got the cuboidal cell shapes, which are like cubes, and the columnar cell shapes, which are like columns. And then also your epithelial tissues are going to be, we can look at them, are they in layers? If it's a simple epithelium, these cell shapes will be just in one layer, like simple squamous epithelium will be one layer of those flat cells. Simple cuboidal will be one layer of those cube-shaped shells. Now that's hard to say. And a simple columnar epithelium would be one layer of those column-shaped cells. If we have a stratified epithelium, then these cells are in many layers, and stratified epithelia is always for protection. Now the pseudostratified, these are false layers, and the only type of pseudostratified epithelium that we have is going to be the columnar type. So we're never going to see pseudostratified mixed with these words up here. So we'll only have pseudostratified columnar epithelium. And then this term transitional. Transitional is a type of a stratified epithelium, and it's really of squamous type. We'll get to that here in a little while. So transitional is another type of stratified. We just don't have that in the word. We're going to start out with the simplest epithelium. On all of these, we're going to go through simple squamous first, and then simple cuboidal and simple columnar. And then we'll hit the stratified epithelia, and then the pseudostratified, and then finally I think we'll get to the transitional. That's going to be our game plan. And so here is simple squamous epithelium. And these are the simple squamous cells going across here. They're very flat cells, and this is in the lung. This is an alveolus in the lung, an air sac of the lung. And so the reason we want to have these flat shaped cells, here's a nucleus of one. These are the nuclei of them. They're just very flat cells going across here. We want these very flat, squish-shaped cells, very hard to say, so we can have diffusion from this alveolus into this. This is a capillary. These are little red blood cells in here. We want oxygen to easily diffuse across here. This is called the respiratory membrane. Here's a little simple squamous cell here. This is a simple squamous cell here that's lining this capillary. So oxygen can easily diffuse across this respiratory membrane into this blood vessel here, and then oxygen can be distributed throughout your body to the tissues. And so the reason we want simple squamous cells, little flat cells, and only one layer here is for diffusion. And so here's another simple layer of simple squamous cells. Here's a simple squamous cell here with this little nucleus, and here's a nucleus. So let's go to the next slide so you can get this search image pattern going. Here's simple squamous again. So these are all simple squamous cells here. All these little flat cells going across here, and you can see the nuclei to each of them. These are also cells that have been sloughed off. So you can see from above that they just look like little eggs. There's a nucleus inside of them. But this is what it looks like inside your lungs, and these are the air sacs inside your lungs. It's called an alveolus, lined with simple squamous cells. Not much more to say here, but I'm hoping you can see this. These are the cells from the side, very flat, squished cells, and in one cell layer thick. The purpose for simple squamous is not only diffusion in your lungs, it's diffusion, but you can also have filtration occurring. Filtration is not going to occur in your lungs, but it will occur on the kidneys, and we'll see the kidneys here in a second. Now these are also simple squamous cells. Here's a simple squamous cell, here's a simple squamous cell, here's another simple squamous cell. So here we're lining a structure, so you can see the very flat cells in here. And notice how the nucleus of simple squamous cells are nice and squashed and flat. Don't worry about all this other stuff out here. These are what simple squamous cells look like. Now, if you look at this picture and try to find simple squamous cells, I'm going to give you a second before I point to them. You try to locate a simple squamous cell on the slide. Can you do it? They're lining a structure. Here they are, right here. This is the nucleus to one, and it extends on around, and here's the nucleus to another. It extends on around, here's the nucleus, and it keeps on going. This big huge thing in here is called a glomerulus. It's a ball of capillaries inside your kidney, and this is where urine is being formed, and the urine is squeezed out into this area here called Bowman's capsule, and Bowman's capsule is lined with simple squamous cells. So this is what simple squamous cells look like in your kidney. Now really the filtration is occurring here in this capillary bed, but simple squamous cells are all in this capillary bed itself, and filtration is occurring. But these are the simple squamous cells here, lining all around the edge. So there is a big nucleus there, and here is a big nucleus here. All of these cells out here are going to be a type of tissue that we're going to run into here in a minute. There's a beautiful example here of cube-shaped cells. These cells right here are simple cuboidal cells. Now see if you can find the simple squamous cells on this slide. They're lining Bowman's capsule again, right here. Here's a big nucleus to one. Here's another nucleus. Here's another one. Here's another one. These are the simple squamous cells here, lining the inside of Bowman's capsule. And here's our big glomerulus, where urine would be formed inside of our kidney. So this is what the inside of a kidney looks like. So those are examples of simple squamous inside the lung and inside the kidney. The job of simple squamous would be diffusion or filtration. Our locations would be the kidney and in the lung. There are other locations in the body, but those are the easiest. Now in this slide, you see these regions here, here, just about everywhere. All these rows here, 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 everywhere where I'm going around like this. What they have done is they have cut through tubules. They've taken cross-section through tubules in the kidney. Also in the kidney, we not only have simple squamous epithelium, we also have these types of cells, and these cells are more cuboidal shaped. This is simple cuboidal epithelium. Now you might be asking yourself, why isn't this stratified? Why isn't this considered many, many layers? And it's because we have an open lumen here, and this is just a single layer. Every single cell is touching a basement membrane here, and then it's open to a lumen. And so this group of cells here opens to a lumen and rests on a basement membrane. This group of cells opens to a lumen and rests on a basement membrane. So you might think, well, this is 2 cell layers thick. It's not really. Each of them has their own basement membrane and opens to the surface. And so all of these are examples of simple cuboidal epithelium. They're little tubes that have been cut in cross-section. Here's a very up-close view of simple cuboidal epithelium, so I can talk about what I was just talking about. So here's a nucleus. The nuclei to simple cuboidal epithelium tend to be nice and round, where in simple squamous they were squished. So here's a nucleus, and here's a nucleus, and here's a nucleus. So this is a tube of simple cuboidal. Here's a tube, here's a tube, here's one, here's one. What I was saying before, if you're wondering why isn't this considered stratified, because here's the basement membrane right here to our simple cuboidal epithelium, and here's the open lumen up here. So here's the apical surface, and here's the basal surface, and then underneath this basal surface here, this basement membrane, we have connective tissue down here. If we had another layer of cells, then it would be stratified, but we don't have another layer of cells. We have connective tissue down here. Here's a very clear example of simple cuboidal epithelium. This is very nice. Simple cuboidal here, simple cuboidal, simple cuboidal. All of these are individual rows of simple cuboidal epithelium. Little, and they look like pearl necklaces. And either you have this and you're like, duh, move on to the next slide, I so see this, or you are ready to punch your computer screen right now. What I would suggest, if you can't see this and you don't understand this, is that you come in and talk to me, and I can walk you through this, and I can draw up on the board. It's so much easier for me drawing this, starting from scratch, what the difference is between the simple and the stratified epithelium. Epithelial tissues, because once you get it, but until you get it, you don't have it. And I'm sorry if you don't have it right now and you just want to come through the computer screen and choke me out. Okay, now those are the simple cuboidals. Now let's move to the columnar. These cells are much taller, and usually, now this is a round nucleus, but usually the nuclei to the columnar are much more elongated. See how they're elongated? And another thing about the columnar cells, see how they're purple on the top here? That's because the stain, it stains much better on the edges, because on the columnar epithelium, these, I should go back. What I didn't tell you about the cuboidal. You find the cuboidal epithelium always in the kidney, that's a given, because these are kidney tubules, and you find them lining ducts. But the kidney is a big one. And the biggest thing that cuboidal epithelium does is secretion and absorption. You can put secretion on your test, or you could put on your practical, or you could put absorption, either or, one or the other. Now when we get here to the simple columnar epithelium, the same thing for the job is either secretion or absorption. This purple lining reminded me of its function, the absorption function, because you find simple columnar all lining the entire digestive tract, from the stomach all the way through the small intestines to the large intestines. And the small intestines is where all your nutrient absorption is going to be. And so we need a really large absorptive area. And so on the simple columnar epithelium, you usually have a lot of microvilli. And the microvilli, what they do is they increase the absorptive area, and they're folds in the plasma membrane. And so when you get all these little folds in the plasma membrane to increase absorptive area, it really takes up the dye very well, and so it stains darkly up here. So these are microvilli on the edges of our columnar cells. Now you see all this stuff down here? This is areolar connective tissue. Areolar connective tissue. It doesn't look like it when we get to later slides, but all of our epithelial tissue rests on connective tissue, and all of our connective tissue that opens up to the outside is going to any connective tissue that opens to the outside of our body, like our respiratory passages and our digestive passages. All of those are called our mucous membranes, and any mucous membrane that we have is going to rest on what's called a lamina propria. So this is a lamina propria. It's just another term for our connective tissue. And that lamina propria is always made of areolar connective tissue. So this is what areolar connective tissue looks like when you don't spread it out and pull it out really tight on a slide. So notice how elongated these cells are. That's a really nice slide of simple columnar epithelium. And usually, like I said, the cells or the nuclei are elongated as well. We can find this, the best place to find this is all along the digestive tract. You're not going to find it in the mouth, and the reason you're not going to find it in the mouth, I'll go ahead and tell you this, you're not going to find it in the mouth and down the esophagus because there's too much abrasion. So you find stratified squamous in the mouth and in the esophagus, but once you get to the stomach, all the way down to the small intestines and large intestines, this is the type of epithelium you find, because this is modified for absorption and secretion. You don't find this in the anus, way too much abrasion there. Here's another slide where we have simple columnar epithelium. And so we have those elongated nuclei, nice oval nuclei, and nice elongated columnar shaped cells. And here it's simple columnar down here too, it's just hard to see. And all of this is the areolar connective tissue. This is our lamina propria that supports our columnar cells. Well, I guess that's it for columnar. We just have those two slides. Now, so we've gone through all of our simple epithelial tissue, simple squamous, simple cuboidal, simple columnar. Now, before we move on to the stratified, this is our pseudostratified. So it looks like there are many layers because our nuclei are in many rows, but this is just one layer from here all the way up to the top. All of these cells are actually touching the bottom layer here, touching the basement membrane. So this is called pseudostratified, ciliated because of the cilia, columnar epithelium. This is a bunch of mucus and other cells being brushed along the top. So here's our cilia. This is pseudostratified, ciliated columnar epithelium. And I actually see mucus cells up here. This is a goblet cell. Here's a goblet cell. Here's a goblet cell. Here's a goblet cell. So these cells are secreting mucus to push up here for our cilia to brush along. So this is pseudostratified, ciliated columnar epithelium from the point where my mouse is going all the way up top. And it appears to be in many layers, but it's just in one layer. This is debris up here that's being pushed along. See the cilia? So we would find this type of epithelium in our upper respiratory passages, like in our nasal cavity and also in our trachea. And what that does is it helps collect stuff like this. Maybe this is up in our nasal passage, and prevent that stuff from going down into our lungs. And so you could, if you want a quick answer for the laboratory practical, what do I put for its function is to secrete mucus, trap debris. And where would we find this? In our upper respiratory passages, or you could simply put trachea. Here's another look of the pseudostratified ciliated columnar epithelium. It starts down here and it goes up to here, There's the cilia, and these are the goblet cells full of mucus. So there they are, full of mucus. And here's another look for it, starting here and going up, and here's the cilia. It's easy to identify if you can see the cilia. And can you see it there? Here it is. Now, because this is in a respiratory passage which opens up to the outside, this makes this type of epithelium a mucous membrane. If you have this plus the underlying connective tissue, that defines a mucous membrane. Epithelium plus underlying connective tissue is a mucous membrane. And so this mucous membrane, this part of the mucous membrane, the underlying connective tissue, would be called the lamina propria. And the type of connective tissue associated with lamina propria would be areolar connective tissue. So this is areolar connective tissue down here supporting our pseudostratified ciliated columnar epithelium up here. It looks like many different rows, but again, this is pseudostratification. This is false layering. All right, do you see something on the slide that you recognize? Up here we have pseudostratified ciliated columnar epithelium up here. And I'll go ahead and tell you that all of this is our areolar connective tissue, and this is going to be something in the future that you're going to learn. This is called hyaline cartilage down here. So the purpose of this is secretion and debris removal, and then we're going to find this in our trachea. So then we'll find hyaline cartilage in our trachea as well. Now we're going to move on to our actual layered stratified epithelium, and this is stratified squamous epithelium, and there are going to be two types of stratified squamous epithelial tissue. There is the non-keratinized version and the keratinized version. This is non-keratinized, and where this starts, you see these little papillae down here. We start from here. These are where the cells are actually, living, and they're reproducing, and then they push the cells all the way up to the surface here. Here's the outermost surface, the part that you would see. This is our stratified squamous epithelium, and it goes from here all the way down. And then this is our underlying connective tissue. This would be our areolar connective tissue. This would be the lamina propria, the areolar connective tissue. So here's our stratified squamous, and the reason we know that this is stratified squamous, when you have stratified layers, down here these cells look quite cuboidal, but you look at the top layer and you see how flattened these cells are. Look how flat that nucleus is, and we name our epithelial tissue by the top layer of the cell, by the apical surface, and so this is stratified squamous. No matter if these cells are cuboidal or columnar down here, we go to the topmost layer and we name it that way. So where we find stratified squamous epithelium, the non-keratinized version, would be areas of the body where there's going to be a lot of abrasion, but they're on the inside of the body, because the keratinized version is going to be on the outside of the body. So this type of epithelium would be on the inside of your body, like in your mouth and in your esophagus. The non-keratinized stratified squamous epithelium. And it could be in the anus, in the vagina, and that's all I've got for you right there. Mouth, anus, vagina, areas of high abrasion that don't need to be waterproofed. Here's another look. It's the same thing. We have non-keratinized stratified squamous epithelium. We're starting down here at the bottom, and we have these many layers, and when we get to the top we have these flat cells. And the way you're going to be able to tell this non-keratinized stratified squamous epithelium from the keratinized version is that the non-keratinized version has nuclei all the way up to the top. All the way up to the top. I don't know if you've ever been in biology class and you've taken a toothpick and you've scraped the inside of your cheek and then put it on a slide and stained it, if you've done that and sloughed off some of these cheek cells inside your mouth, you can stain them, you'll actually see nuclei in those cells, and that's because the nuclei are all the way up to the surface. Whereas if you did that with your skin cells, you're not going to be able to see those nuclei because they're down quite a bit deeper. So here's non-keratinized stratified squamous epithelium, non-keratinized stratified squamous epithelium. Here's that areolar connective tissue underneath. And here's another picture of the same thing, non-keratinized stratified squamous epithelium. This could be a vaginal sample, this could be a sample from the esophagus, from the mouth. It's still stratified squamous epithelium, and I know it's non-keratinized because we have nuclei all the way up to the top, and when you see the keratinized it looks completely different. So here is our areolar connective tissue down here at the bottom. Now this is what keratinized stratified squamous looks like. Here is the non-keratinized, here's keratinized, but it's up very close. This is what your skin looks like. If we look up close, this is way too close of a picture, but on the outside of your skin, the outermost layer of the stratified layers, you don't see any nuclei. You just see dead cells. They just look like dead cells that we get further away. Here is that layer. This is kind of a real far away picture. This is the layer right here, and I'm outlining the bottom of the layer of stratified squamous epithelium, and the layer will go from here all the way out, like here, from here all the way out. And so what this stuff out here is this is the dead outer portion of your skin that flakes off. You can think of it as dandruff or dry skin. So from here out, or from here out, what this is stratified squamous epithelium, but we would say that this is keratinized stratified squamous epithelium. It looks way different, doesn't it? And you're thinking, well, it doesn't look anything like this slide. It doesn't. From here out, this would be keratinized stratified squamous epithelium, but the difference is these cells right here are compact and they're not scaly like these. They are not ready to shed. So this is the outer portion of your skin right here. This is called your epidermis. That's your epidermis. All this stuff right here we're going to get to when we talk about connective tissue. It is your dermis. It's the underlying dermis. I wish we had a closer version of that. That would be helpful. Maybe we will later on. Now this is transitional. This is a type of a stratified epithelium because you do have many layers and they're starting here and going all the way up. But the thing is we can't call it stratified squamous because see the top cells, they're not squashed. And we can't call it stratified cuboidal because they're not so cuboidal, and we can't call it anything like that. We call it transitional because this type of stratified epithelium, it goes through transitions. We find this in the bladder and in the urethra and in the ureter, mostly in the ureter and the bladder. But what's going to happen is when your bladder is empty, then this transitional epithelium is going to look like this. But when it gets full, your bladder gets full, this epithelium is going to stretch and it's going to look more like stratified squamous epithelium. So it's going to go through transitions. The way you identify this is it looks dome-shaped. So you can say that its function is distensibility and its location is the bladder. Did I give you a function on this one? Functions for both of these guys, for all the stratified squamous, is going to be protection. Location for this one, which I did not give you, was the skin. The keratinized stratified squamous epithelium. Forgot to give you that. So there's for our transitional. To figure out transitional, look for dome-shaped cells at the top. See these big puffy cells? Then you'll know you've got transitional. Here's another transitional. And transitional, just like those other ones, they do have a regular connective tissue down here, and then they're going to have smooth muscle down lower. But these are those dome-shaped cells at the very top. And then you know you've got transitional as soon as you see these little puffy cells. And our transitional goes from here, up, many layers. And so it is a stratified epithelium. It just goes through transitions. And there's another transitional. That looks ridiculously familiar. Okay, dome-shaped cells at the top. And we have our transitional. So distensibility is the function, and location, good location would be the bladder. So we'll stop it there.